I don't find solutions. So I'll talk about those problems today. Should I go ahead and try to share my screen and all that?
Yes, go ahead.
All right, let's find that one. Did that work?
Yes, probably you should put full screen of the presentation. So
I thought I did. Oh, there we go. Okay, it finally worked.
okay,
I'm going to
that's
grab a
perfect
laser
now.
pointer here, and I thought I was going to. Excuse me. And I'm going to get started. All right. So thanks again for dragging me out of bed at this hour. Actually, I usually get up at this time anyway, so no big deal. So I'm going to talk about stress and exosomes and GBM and potential therapeutic resistance. I got nothing to disclose. I've got a lot to cover, so I'm going to kind of race through things. I'm going to talk about way too much and way too little time. The things I won't talk about, excuse me, I will not talk about stress and exosomes and immune suppression or immune stimulation. We've been on both sides of this coin, and this is a big issue, but I'm not going to discuss any of that today. I will not talk about also the sort of subcellular, extracellular location of chaperone proteins or heat shock proteins. That's another really interesting area, but not going to cover any of that. Here's the vague outline. So I'll talk a little bit about glioblastomas. Most of this crowd probably already knows that. I'll talk about exosomes, or also called extracellular vesicles. I'll talk about the stress responses, in this particular case, the unfolded protein response, and they'll combine everything. Stress GBM exosomes, or what I call sexosomes. I tried to get that through our patent and trademark office, and they threw me out the door. So it didn't work in my hands, it may work in yours. Glioblastoma. Okay, this is the most, we obviously call these things these days. Glioblastoma, grade 4 astrocytoma, IDH wild type, and then all the listings of the various genetic changes that may go on behind it. I won't talk about that. But this is the most common and unfortunately the most deadly of the brain tumors. As most of us know, this is a 15-month or less median survival. We're kind of improving that a little bit, but not much. And this has been about the same for 20 years. So we've really hit a wall with this, and it's almost uniformly lethal. This picture here shows pretty much everything I'm going to describe, that, and some things you can't tell. This is an extraordinarily heterogeneous batch of cells. It's kind of believed that each cell is actually different from every other cell in there. Obviously quite proliferative. It's covering a huge mass in the brain. Very, very invasive. It's crossed the corpus callosum. That's kind of a surgical no-go for most neurosurgeons. It's extremely vascular. You can see how bloody it is. And there's this hypoxic core here. And there's other areas of extraordinary hypoxia throughout it as well. And the main thing that we're going to talk about today is that it's resistant to therapy. And our main therapies are surgical resection, combination of chemoradiation, and then we're left with experimental therapies. The obvious therapeutic difficulties are that it's in the brain. So you can't really suffer collateral damage. You can remove a breast, or you can remove a kidney. You really can't remove a brain. It kind of messes things up. The blood-brain barrier is a massive issue in trying to get drugs into the brain. The immune system, I'm not going to talk about the immune privilege site that we call it, but that's an important aspect of it as well. And because it's in the brain, it can have any other CNS downstream effects. It's not, we see patients often where they end up having a stroke from their tumor. So this can obviously lead to collateral damage. So I will talk about stress. And man, we're all so stressed. Everybody's stressed. Why are my cats so stressed? I don't know. And stress, on the other hand, can lead to resistance. You can overcome stress by essentially fighting it off and becoming stronger and nastier. And that's what tumors do. Tumors are existentially stressed, either of their own making, because they're so metabolically active. They undergo oxidative and hypoxic stress, ischemic stress. They need nutrients that they can't necessarily get. And the stresses we impose on them as we try to treat them, often, I work in thermal areas, so focused ultrasound and laser ablation, but just the other therapies that we use also lead to these sorts of stresses, which often lead to proteinopathies or foldopathies. Okay, now quickly about extracellular vesicles or exosomes. We use the term extracellular vesicles because exosomes has kind of gone out of favor. I'm an old person who likes the old terms. I'll continue to use that. And hopefully some of you are familiar with this, but I'll try to go through it quickly. An exosome forms through an endocytic event. It starts out at the cell surface, forms an endosome, goes through the endosomes change and make little vesicles inside a big vesicle. This fuses with the membrane surface, and out come those little vesicles. And that's what they're called. They're called exosomes, and they're about 30 to 100 nanometers, so kind of virus-sized. Other means of getting vesicles out of a cell is to pop it directly off the cell surface. We call these microvesicles and other various terms. And these are usually larger than these, but not necessarily. And since we can't tell the difference, we tend to call them extracellular vesicles, but I'll continue using the term exosomes. There are other ways to get things out, and they will release vesicles as well: secretory lysosomes, autophagosomes, and non-canonical secretory autophagosomes. And we also now describe other kinds of tiny little particles that come out. Basically, if you can centrifuge something hard enough, you can find something new, and that's kind of where we've gone. So you can see this is a very lovely drawing, and I tried to submit this as in a review article, and one of the reviewers was concerned because of this that I actually had a stroke. So they had an actual artist draw this, and this is what it came out looking like. But we draw these vesicles as kind of large things to emphasize them. The fact is they're actually quite tiny relative to the size of the cell. Again, they're on the order of viruses. So, Chloe, please, I'm talking. These things are viral size and obviously hard to see. They'll hold about 100 proteins on the inside, maybe 200 on the outside, and several thousand nucleotides worth of stuff. But so they're quite small. They don't hold a lot, but in a swarm, they hold a lot of material, a lot of information. We kind of regard them as God's gift to science. And I realize with the number of potential Italians on here, this may not have been the best slide to show. What do these mean in terms of tumor biology? So if we break it down into tumor cells and normal cells, what happens? Tumor cells secrete these vesicles often to help themselves. They're very good at, they can actually passage aberrant signaling molecules in brain tumors. For instance, EGFRV3 can be passed from cells that have it to cells that don't have it. Those cells will take up the vesicles, actually display EGFRV3 on their cell surfaces and induce all the downstream signaling pathways that go along with it. We know that they can maintain a stem cell niche, which is one of the most resistant of the niches. It can also change the sort of grade or the nature of the tumor from say a proneural to a more of a mesenchymal. And I'll talk a little bit about stress conditioning. In addition, they can literally fend off things physically just by being in the way. They're very angiogenic. They drive blood vessel formation around the tumor. They invade into the normal tissue, and it can actually co-opt this normal tissue into aiding and abetting the tumor. If these were to be metastatic, which most brain tumors are not, they will actually, what I say, refer to as setting up the pre-seed in soil. These will actually set up the niche for metastatic tumor cells to come and find that new location. They're actually able to package drugs and toss the drugs right out of the cell. So drug effusion is one thing, and I'll talk a little bit about drug resistance in terms of metabolomics. Once outside the cell in the periphery, they are actually very good at immune suppression, and they can serve as immune decoys. And one of the things we love to talk about, but don't see much of it happening, is that we think there might be good biomarkers. Well, that's not part of today's story. We have a wide variety of ways to isolate and characterize these. I won't go into any of this, just say that it's able to be done. The thing in the field is no one's going to like however you do it. So we just sort of accept that and carry on with your life. GBM exosomes and therapeutic resistance, what most of the studies have shown are involving the various RNAs that are carried by these things: microRNAs, long non-coding, and circular RNAs, usually in the context of temozolomide resistance, but also in terms of radio resistance or X-ray resistance. So these are factors in that, also just changing the nature of the cells themselves. And there's more and more stuff. If anybody wants this talk, just let me know and I'll send it to them. We've found a couple of other ways where GBM vesicles actually cause neurotoxicity. So this is not necessarily a resistance phenomenon, but a danger phenomenon. This is very disconcerting to me, and we're trying to find ways to inhibit that. Excuse me. From the stress side of things, I see stress as a protein folding problem. You can have proteins that are malfolded as they're being generated in the cytosol. Those can also lead to inappropriate interactions, and these two things can combine for essentially to make a dangerous and essentially deadly situation for a cell. And that's why you have chaperones to try to prevent that from happening. If these unfolded events start happening in the endoplasmic reticulum, you have kind of a different story than you do in the cytosol. In the cytosol, we call it the heat shock response. In the endoplasmic reticulum, we call it the unfolded protein response. And here are a bunch of words. Let's not go into that. Let's first talk about what normally goes on in the endoplasmic reticulum. Proteins are made in the cytosol, are being generated in the cytosol via the ribosomes. If that protein that's coming off the ribosome has a signal particle or signal peptide on it, it'll be recognized by what's called a signal recognition particle and dragged into the endoplasmic reticulum, where it's now translated inside the ER. Once inside the ER, a lot of things happen. It can get glycanated, it gets carbohydrates added, it gets folded, and disulfide bonds are formed. And this requires ATP and calcium to be done correctly. If things don't go so well, then the protein is considered not fixable. It could be tossed out through what's called ERAD, endoplasmic reticulum associated degradation. So it goes into the cytosol and is chewed up by the proteasome. If this happens too much and you can't control it, it induces the unfolded protein response over here. Now, we have chemical tricks by which we do this to cells. One is to stop glycosylation by using tunicamycin. Another is to allow calcium release into the cytosol with apsigargan. And another is to block the disulfide bond issues here with dithiothreatol. In our hands, we've used all three of these to inhibit or to induce, I should say, the the unfolded protein response, most of which we focus on DTT because it's cheap and easy, kind of like me. Quickly, what is the UPR in terms of in a nutshell? So as unfolded proteins accumulate in the endoplasmic reticulum lumen, GRP78, a chaperone, also called HSPA5, is hanging on to certain proteins, PERK, IRE1, and ATF6, but it has to let go of them and go start trying to deal with these unfolded proteins. This allows these proteins to do different things. One, it can escape into the Golgi, or these can actually dimerize or oligomerize, and a lot of downstream things happen, most of them involving a translational block, a change in the transcriptional landscape. The proteins that are allowed to be produced actually can be done so through internal ribosome entry sequences, so they can avoid this translation block. In the end, what you end up doing is finding that hypoxia and glucose starvation cause this in real tumors, not so much in the tumors that we deal with in cell culture. But the downstream effect is, how do we fix this? And one is to fold things and fold the proteins so you make more chaperones. If you can't stop that, then you end up killing yourself. The cell will undergo apoptosis through a couple of different means, but also it hedges bets to try to return things to normal in case it can overcome this. So the translation block goes away, proteins get made, things like that. Basically the same emotions we all feel when we get a bad grant review back or a paper review back, cells do the same thing. So that's kind of that in a nutshell. Stop translation, degrade RNAs, and change the transcriptional landscape to make more chaperones. We've published a number of papers on this sort of thing. I'm just going to summarize a lot of that data. We've used cell lines, we've used xenografts, we've used primary cultures, in other words, things right out of a patient's surgery, and primary cell lines that we generated as neurospheres, and we've used the tumors themselves. In every case, what we see is an increased chaperone content compared to normal tissue, and we can upregulate that chaperone content by stressing those cells even further. And what we'd often see is this is a metabolic phenomenon. Just quickly to show this is a bunch of Western blots showing all these endoplasmic reticulum and UPR type stuff. And this is brain over here. So we don't see nearly as much of that as we do in the tumors. That's just throwing that out there. The one thing that really disturbed me in one of our experiments, though, is if we follow translation over this time, so we induce the unfolded protein response and translation drops like you would expect, but then it just turns right around in the midst of the stress, and the cells continue to produce proteins, and that's seen as a dephosphorylation event for EIF2 alpha. So the tumors don't really care. They'll just push right through this, and they'll probably use it as an adaptive means. One area, as I've mentioned, we kind of followed is the metabolism of this, often in terms of the Warburg effect, but I think it's more than just that. And to quickly show that, if we induce stresses in U87 cells and we follow what's going on with labeled glucose, we see an increase in lactate, we see an increase in glycolysis, and we see hallmarks of tumor aggressiveness, increases in alanine and UDP glucose, increases in phospholipid synthesis in particular. These are predictive of drug resistance, and other people have shown that besides us. We've also seen this in tissue slices, tumor tissue slices, so it's not just cells. And if we look at the tumors themselves, and particularly recurrent tumors, these are by definition treatment resistant. We see a change in the metabolism towards a lipid production, and we also see sort of a change in the unfolded protein response leading toward a particular pathway, the ATF6 and XBP1. Bear in mind that when you're making, when you need to get rid of protein through the secretory system, you're going to have more lipids, so that makes sense. And with more lipids, you also end up with probably more vesicles, and I'll get to that in just a second. We've shown that the enzymes of lipid biosynthesis and glycolysis are somewhat upregulated, not ridiculously upregulated, but that seems to be the case. Now, the question is, does this lead to a therapeutic resistance? And yes, it does. So these are U87 cells happily growing in culture. We treat them with temozolomide. They're kind of resistant, but their growth does go down. If we induce the unfolded protein response in those cells, they grow faster than normal. And if we induce that unfolded protein response and then try to treat them with drugs, it bounces right off. So temozolomide, the drug of choice, just has no effect in this situation. Using other tumor cells, these are primary lines, and treating with an HSP90 inhibitor that affects one of the chaperones of the endoplasmic reticulum, the same event occurs. The cells grow as fast or grow faster if we induce the unfolded protein response. If we treat with drug, it goes down. If the unfolded protein response is engaged and we treat with drug, it bounces right off. The cells return to normal. So this is kind of a disturbing event because also the drugs themselves will induce this unfolded protein response. So we're basically making the tumors become resistant to their own, by their own mechanisms. Other things we've shown exposing tumor cells to their own exosomes is changes in proliferation, metabolism, invasion, and migration, and a lot of changes in signaling. And if these are stressed vesicles, those signaling changes are even more exacerbated. I'm required by law to show how we separate vesicles, and this is how we obtain them from cell culture. We don't have to spend too much time about this. These are just ways to check for the various components that we think are in them. This is a size and concentration measure. These are what the cells look like. We grow them as neurospheres, so they grow in these big fuzzy balls, and they're just kind of disturbing looking to begin with. If we induce the unfolded protein response, we see 3 to 5 times more exosomes released from these cells. And it doesn't matter what cells we use, and it doesn't matter who purifies them. And we see this both in protein and what's called vesicle number, or nanoparticle tracking analysis methods. So we see this in both ways. The cells make more vesicles when they're stressed in this particular manner. And so here's the basic way we look at things. We have cells sitting in the dish. We do nothing to them, or we induce the stress. We take their exosomes and passage them to other cells and then ask, what do we see in the end? And we've done proteomics on this. It looks like it induces an unfolded protein response phenotype, but it also induces that metabolism change, that increase in glucose uptake and lactate release, which is sort of indicative of glycolysis, and increases in lipids as well. So pretty much the same things we see if we induce the unfolded protein response, we see if we induce the unfolded protein response by throwing stressed exosomes on the recipient cells. And that's just kind of shown here more along that same idea. The protein changes are quite evident between cells just sitting there and cells that have had the unfolded protein response induced, or cells that are treated with exosomes, or cells that are treated with stressed exosomes, those that came from cells that had the unfolded protein response induced. We've done a ton of Western blots to show that this actually occurs at both the chaperone level, the transducer level, and the signaling level, and it just kind of goes on and on from different cell types just to show we've done that. How do we analyze this? We tend to use ingenuity pathway analysis at the insistence of our biostatisticians. What we can see from that in a big picture sort of assessment is that the unfolded protein response and exosomes of the unfolded protein response induce the cells to look about the same. The pathways and the diseases and biofunctions are quite similar to each other if you just look at that list of categories there. If we look at the differentially expressed proteins in what's called the comparison analysis, the heat map shows some pretty strong similarities between the two. So the vesicles kind of make it look like they've stressed the cells in the same way that the stress does. That can mean that an unstressed cell receives stressed vesicles and suddenly takes on the stress phenotype. And remember, that stress phenotype is usually going to result in chemoresistance. What about the exosomes themselves? What are the proteins in there? Well, looking at the big picture again, we see a lot of signaling pathways that seem to be involved with the EVs that come, or the exosomes that come from stressed cells. And that impact is shown on the recipient cells. These are cells where we just induce the unfolded protein response. We see these changes in these various signaling molecules, and this is phosphorylation events. If we take stressed vesicles and put them on the cells, we see similar, to some extent, but an increase in these signaling events via phosphorylation. So there is a signaling phenomenon, and it also goes to the receptor tyrosine kinase level. The others were sort of intracellular kinases. Does it protect things? And well, yes, it does. So that probably wasn't going to be a shock. If we have cells growing in culture and we induce the unfolded protein response, as I mentioned, they grow faster. If we add exosomes to them, they grow faster. If we treat with drugs, it knocks them down. If we treat the cells with exosomes, whether or not they're stressed, and then treat with drugs, it bounces right off. So the vesicles can transmit this sort of chemoresistance as well. I'm going to wrap this up with the RNA work. We've actually taken the RNAs from the vesicles, essentially repackaged them in liposomes and threw them back on the cells. And in the big picture of things, looking at all these canonical pathways and using hierarchical clustering, this is the unstressed side of things. This is the stress side of things. So you can see just by looking, there's sort of a stress event induces changes in a number of these canonical pathways, no matter how you delivered it, whether it was direct from an unfolded protein response induction or with vesicles or with the RNA from those vesicles. And I'm going to point out some pathways because you can't read any of this. I can't read any of this. We see ERK signaling is upregulated, PI3K AKT signaling is upregulated, AMPK signaling, hepatocyte growth factor signaling, EGFR signaling, ephrin receptor signaling, and glioma invasiveness signaling. All these things are critical to glioma pathology. So this is something that's very frightening to me, is that we stress cells, the cells take the stress vesicles, throw them on other cells, stress those cells, and now you've got a more, essentially an angrier tumor than you might have had before. Can we do anything about this? And this is where I get into the problem. I find problems, I don't solve them. There are people trying to modulate so much the unfolded protein response. There are a number of things out there. None of these have reached sort of a clinical stage, mostly because they have a lot of off-target events. Same thing with trying to inhibit extracellular vesicle formation. If you can stop the cells from making vesicles, can you stop these downstream issues? And I would argue that's difficult because cells like to make vesicles. This is what they do. And also these can have a lot of downstream off-target effects as well. Can we block the EVs? This is something we're trying to do to figure out how to do this in the tumor microenvironment. Trying to remove the EVs may have systemic effects, and that may be good, but that's also, it's been pretty hard to do. So right now, I don't think we have good ways to overcome this. So to wrap this up quickly, the UPR is ostensibly a stress response, but tumors and gliomas in particular kind of like it. And they use it to increase their proliferation, to protect themselves from chemotherapy, possibly through metabolic changes. And a lot of this, I think, relates to lipid metabolism. The vesicles released by them can actually pass this stress phenotype to recipient cells. And we see the same sort of downstream things, increases in tumor aggressiveness and therapeutic resistance. And some of these pathways, on the other hand, may represent targets, but we haven't quite figured out how to hit those yet. And yeah, the RNAs, I didn't go into that, but they're probably quite important in this. So to wrap up with the people who actually did the work, here they all are. Here's the money. I want to thank the money as well. My brilliant PI level collaborators. And thank you for your attention. I hope I stayed close to time.
Yes, thank you very much. That was really interesting and really complex presentation.
Yeah, sorry about that.
No, it was really interesting. Maybe I can start the session with the questions. Are there any questions? Maybe yeah, I start at the beginning. I'll say, sorry?
No, you can go ahead first. That's okay.
Okay, I was enjoy a lot the part of the unfolding protein response, because I was working also a little bit with that. And really? I was thinking, have you ever induced this unfolded protein response for the longer time? Like, because I see the incubation for 4 hours or for 24 hours, and I don't know. As I know, it's like normally when you induce unfolded protein response, first the cells try to protect themselves and they activate all the pathway to survive. And then if it's longer, more heavy and prolongated, they start to die because they are not able to compensate.
That's a great question. We've not done it in that respect. For one thing, dithiothiazole has a limited half-life. So that's kind of why the 4-hour experiments, these are typically done. Now, have we sort of continued to do that? Have we kept adding that? We've not done that. And that does beg the question, what would happen? Now, from a real tumor perspective, the stresses are essentially continuous to some extent, right? So my thought would be that in an actual tumor setting where you've got all the necessary cells of the microenvironment as well, that you'll actually create a more aggressive tumor. I think in culture, you'll eventually kill things. because we can, they don't necessarily have the other support mechanisms to increased angiogenesis, neighboring cells that are not so much affected by the treatment, things like that. So I would argue, now I don't know that, but that's my take on what we see when we plot recurrent tumors, for instance, compared to the primary tumors. But from a cell culture perspective, that would be something, we haven't done that because it's difficult to do it correctly, at least in the setting we did. Thapsigargan and tunicamycin do last longer. And we see basically the same thing. You usually treat them for 24 or 48 hours.
Okay, no, because we also see some experiment that prolongated also the exposure of this agent. Later, of course, linked to the other mechanism of the resistance, like all the treatments, more or less. That okay, thank you.
Yeah, well, one thing I wanted, so one of the sort of apoptotic inducers is CHOP or GAD154. This is meant to, it induces a lot of downstream events that lead to apoptosis. One thing we've noted, and I don't know, we haven't tested this extensively, but we did look at a pretty broad array in tissue culture, or in immunohistochemistry rather. We saw that CHOP did not seem to go into the nucleus. It seemed to be retained in the cytosol. So there's potentially even another mechanism for avoiding those, even though the apoptosis should be triggered, it wasn't coming on. And there may be other mechanisms that lead to that prevent, for instance, get the accessibility of these transcription factors into the nucleus. So that even then we're looking at another stage of danger, fear.
Yes, but CHOP is a difficult protein also with the Western, or that happens sometimes that it's easier to see the caspasis, in my opinion, but.
Right. And well, obviously the cells aren't dying. So, but that's just going to throw that out as another sort of way that it's possible to prevent the apoptotic cascades that should be going on in theory.
Yes. Okay. I think.
Yeah, I was gonna question. Yes. Hi, Michael. That's a great talk. Thank you for sharing your data. It's exciting. My question to you, do you know what signals that protein get that package into this exosome? Is there any specific signal, or it is a random event that the packaging everything during stress? There must not be a random, yeah?
Okay, that is something that we've really not studied, but want to understand is what is the change in the stress loading, so to speak. We don't understand cargo loading in vesicles pretty much at all. So it's been regarded as random. I don't think anything's random. But I do think that we don't understand the cargo loading mechanisms. One thing I think is going on is there may be a packaging of degraded or semi-unfolded proteins into these as a mechanism of getting rid of them. This is actually, exosomes were first discovered as garbage bags, getting rid of the transferrin receptor in a situation where you couldn't degrade it. So I think that's kind of been a forgotten element of these. But to be honest, we just don't know. We have a better idea of what RNAs might get in based on sequences of the RNA and based on the RNA binding proteins. But as far as the protein loading, we really don't have a great idea of that at all.
But what is the RNA? I mean, is there RNA modifications that leads to trigger this packaging? I mean, or it is a sequence?
It seems to, well, I would argue it's both ways. People, when we find the specific ones, we're all excited about it. So we know that certain HNRMPs, for instance, will bind certain RNAs and definitely load those. And then we have all the others that we don't know how they got in there. So, and to me, it seems like the RNA story is more random than the protein story. No one ever seems to find the same RNAs, but everybody finds the same proteins. So what has been studied has been the specific RNA sequence that's recognized by a specific RNP, in this case, usually an HNRNP, that seems to get them loaded. From the stress side of things, we don't know anything about that at all. And the other aspect there is a lot of those may be degraded RNAs or chopped up RNAs, because that's what's happening during the unfolded protein response. They're trying to, there's a phenomenon called RID. What's it stand for? RNA, IRE-induced RNA degradation. And so that's another aspect that one of the IRE1s, it sort of degrades RNAs while the unfolded protein response is going on to try to keep the RNA level down. The question is, do those also get loaded in the vesicles, and can those have downstream effects as they're sent to other cells? Because if you got double-stranded RNA showing up, that can almost induce a viral type response, sort of an internal innate immune response through RIG-I and MDA5. But to be honest, we haven't studied this, and I don't know that anybody has. So the loading of these things is still a mystery. To make a hugely long story, that didn't give you any answer whatsoever.
And then you know that when you add those exosomes, you're showing that it resisting therapy like TMZ. What is the mechanism?
We specifically don't know. I again think it's an induction of a metabolic change, particularly because, as I sort of described very quickly, the metabolic profile, the changes especially in the lipid profile, but also UDP glucose, increases in alanine, increases in lactate, and increases in glycolysis. These all seem to be associated with cells that are resistant to not just TMZ, but other chemicals. Some of the kinase inhibitors, for instance. So I would argue it's a shift in metabolism, probably driven through some of the signaling changes that I also kind of alluded to very rapidly. So I think you induce signaling changes, the signaling changes induce metabolic changes, the metabolic changes induce the chemotherapy resistance. And I don't have a specific mechanism for that, just the overall profile of things that we looked at resembles the same profiles people have seen in other systems where they've shown drug resistance involving metabolism.
Thank you.
May I also ask the question, have you checked for the simply ABC transporters in your vesicles? Because that P-glycoprotein is very expressed in glioblastoma, and that could also bring the resistance to...
Right, so I didn't show that here. We've seen that in Western blots. It wasn't the greatest stuff we've ever done. And then, of course, trying to get this approved, and as a grant, they hated it. I don't know why. I thought it made perfect sense. But we do believe, so other people believe anyway, that loading of, say, carboplatin into vesicles occurs because of these ABC transporters. And so not just to the outside, but even sort of into the vesicle, and then you throw the vesicle out as well. So there seems to be multiple ways to do that. And yeah, these transporters do show up in these vesicles.
Okay, thank you.
Sure.
Are there any other questions from the public? If not, thank you very much, Michael, for your presentation. And maybe we can pass to the next speaker, Professor Ahmed. I don't know if I pronounce correctly your name. And
Oh, that sounds perfect. Thank you. Yes, Michael, if you could unshare your screen, please, and I can share my
okay,
screen.
yes.
Oops, stop sharing. There we go. All right.
All right.
Thank you. All right.
No problem. All right. Thank you very much for this, invitations, and morning science is always the best science. Your brain is fresh and you think about a lot. So my name is Atiq Ahmed. I'm an associate professor at Northwestern University, Chicago, and we've been working with this idea of chemoresistance for a long time in GBM. I think Michael did excellent review on GBM, so I'm not going to go into that. But these are the people done this work, especially incredible undergraduate Ella and Jack, who moved on already from the lab in a greener pasture, but they did a tremendous job of driving some of this project and data I'm going to share with you, and of course the funding agency. So as I said, Michael did a good job, so I'm not going to go through this. It's a very deadly disease. One thing that I want to mention is that the origin of these disease, yes, it's still a controversial topic, that where this tumor coming from. And it's considered that they're coming from astrocytes, but in at least in the animal model, when you transgenic animal model, when you induce oncogenic activations in our tumor suppressor inactivations, you can see both neurostem cell and astrocyte can give rise to this tumor. So it's still, we are not sure where it's coming from, but one of the things I want to mention is that probably neurostem cell is driving this the gliomagenesis more pronouncedly than the astrocyte, but astrocyte also can do that. So still, it's a controversial topic, and we are still trying to figure this out, what's going on. So in the nutshell, if you think that why GBM so difficult to treat, I think Michael mentioned this, but I want to re-emphasize this this issue, that why GBM is so difficult to treat. So one of the things is blood-brain barrier. Not every chemotherapy can cross the blood-brain barrier, so we have very limited arsenal against this disease. Second thing is immune microenvironment is a very immunosuppressive microenvironment. And some of the PD-L1, PD-1 checkpoint blockade that work in other cancer is not working for GBM. It is a huge topic, and tremendous effort has been invested on this idea of how to activate this immune microenvironment. Second thing, as previous speaker mentioned, is that it's incredibly heterogeneous. Not each cells are equal in the same patients. So what happened that they don't respond to therapy equally. So you end up having very difficult problem where that you kill some population, but other cells stay alive and then give rise to this recurrence. And finally, therapeutic resistance. These cells are incredibly adaptable, and I'm going to talk about a mechanism we think that's playing a role in this adaptability. So chemotherapy, we work in, we use this as a model, temozolomide, because this is the standard of care for GBM. The Roger Stupp, who was in Switzerland, University of Chicago, Switzerland, now in our institute, who kind of established this temozolomide as a standard of care. So temozolomide damages this DNA by creating this adduct and these 3 adduct. You can see the percent of adduct they create. But what is interesting thing I want to point out is that this 5% adduct that create O6MG drive this effect of temozolomide, anti-tumor effect of temozolomide. And cells find a way to remove this adduct, MGMT, one of the enzyme that's been produced by the cells that can remove this adduct, and this serve as a biomarker where MGMT methylated promoter, where they cannot express the MGMT, patient can survive longer. Okay, MGMT unmethylated one, the patient's GBM progress very aggressively. But what is interesting thing is that regardless of this MGMT status, patients eventually circumvent with this disease. So saying that there's alternative mechanism of resistance, such as Michael showed, and I'm going to show you another mechanism of resistance. So the idea of this glioma is a 2 model that has been proposed. One is that this stochastic model, where it consider as each cells within the tumor are equal and they give rise to tumor. Okay. However, there's a recent model that gained a lot of attentions, which is the cancer stem cell model. This is a hierarchical model where it's considered that within the tumor, there is a stem-like cells that is the main culprit and considered to be driver of this gliomagenesis. And it has been shown that this glioma stem cell has a self-renewing capacity, high DNA repair capacity, and also express high level of efflux pump in their surface, so they can pump out this chemotherapy. And the non-glioma stem-like cells shown here is I don't know. Can you see my arrow? Yes. Maybe I can put it.
Yes, we can see.
Okay. So a non-glioma stem cell, which is rapidly dividing cells, can be targeted by chemo and radiation. So you get rid of this. These cells stay silent, so they can again give rise to these differentiated, highly proliferative cells and initiate recurrence. So now, one of the things that I observed when I was a postdoc, this is my postdoc project. I did it, I had another project that I'm doing on from PI, but this was my observation, and what we were able to show is that observed that under chemotherapy or temozolomide stress, the cells start to become stem-like cells. Okay, so this is a neurostem cell culture in the neurobasal media. And if you put them in without neurobasal media in the temozolomide, they start to form like ball after 6 to 8 days later. So one of the questions I was wondering is that how these cells behaving like stem-like cell. So we generate this reporter system where you have RFP gene driven by this glioma specific promoter, and we express this, and we stably transduce these cells, and you can see that these ball-like cells or neurosphere-like cells under temozolomide express this gene, activate this gene promoter that drive this glioma stem cell niche. So one of the questions that I ask is that whether these cells are converting or expanding within the glioma stem cell populations. So this is a very controversial topic of marker of glioma stem cells because it is very, very plastic. These cells are extremely plastic, so they change the marker. One of the marker that we are using for marking these glioma stem cell is combination of CD133 and CD15. Both of them are surface marker. And if you take these negative cells that don't express this glioma stem cell, we ask the question that what happened when we take these negative cells and expose this temozolomide. And you can see that we sort the cells out, and the double negative cells, you can see day 2 post sorting, they're very, very low, and the yellow bar is a control, and black bar is exposed to temozolomide physiological dose, which considered to be about 50 micromolar. And what you see is that after 8 days, there's a huge expansion of these double positive cells. Yes, but what is to our surprise, what was really surprising, this double negative cell becoming the not double positive cell. So that is the core topic that I'm going to discuss going forward. What we're showing here is that there's a 2 possible way of expanding glioma stem cells. Yes, one is selection, where you deplete this rapidly dividing cell and expanding this quiet quiescent cell. Second thing is that expansions, where you selectively expand these glioma stem cell populations. But our study throughout last 8 years proposed a new possible mechanism where there is a non-glioma stem cell, rapidly dividing cell under therapeutic stress, can expand the population of glioma stem cell. So this is now, when we try to propose this idea, there's a huge push against it because everybody thought that there's a static populations of glioma stem cell, and they liked the idea because they thought that it could be druggable. But we have shown over and over again that these cells expressing this reporter system come in and out of this glioma stem cell state. And we can track this in vivo in the animal model. And you can see that these red cells, this is a SOX2 promoter, red cells staying very close to, perivascular niche, close to the vasculature that has been proposed, that in vivo niche for this glioma stem cell. And what is interesting is that these, when you sort them RFP positive cells, you can see that they are expressing, or they can form tumor when you engraft them very, very small amount. Here we are implanting 100 cells, and 9 out of 7 animal develop tumor, whereas CD130 positive cell, 100% cell express, CD133 also 3 out of 3. So they're saying that this RFP positive cell do possess this tumor initiating capacity. And what is the most important thing that I want to show you, this is probably, how can you prove that in clinically that this is relevant? Yes, we are still trying to do that, but it's very difficult question to do clinically. But at least in animal model, what we show is that if you put a suicide gene driven by this promoter and you deplete this during therapy, what you see is that this is a TMZ plus glycocycovera, where you're depleting these cells during TMZ therapy and you expanding the animal survival as compared to just TMZ. So what telling you is that this is a mechanism that may be possibly driving this recurrence and resistance. Now, why this is important, okay? So as Darwin say, it is not the strongest of the species will survive, it is the most adaptable. So what we believe that this plasticity, where glioma stem cell in and out of the state of different conditions, can drive this adaptability and chemoresistance. I think that's why we think that this is a very, very important topic to target this plasticity, because this plasticity make these cells a moving target, which will be very difficult to hit with any therapy. So last 8 years, we try to understand the mechanism, how this is happening. And we really believe that it's a transient mechanism. Let me see, I'm on time. I have probably 2-3 minutes left. Let me see, I can show you some data mechanism, how is things happening. We really believe that this is epigenetic driven mechanism, okay? What happened is that epigenetic barrier from transition from glioma stem cell state to differentiated state is very high. But during therapy, somehow this barrier is lowered, so they can transit between this state very, very quickly. And we have shown that PRC2 complex EGS2, which deposit the K27 trimethylation, open and closed chromatin, driving these adaptations, okay? When you block this EGS2 activity by drug, you can block the expansion of this glioma stem cell-like cells. And it has an in vivo therapeutic efficacy, but what is the problem is that, at least in our hand, none of this EGS2 PRC2 complex can penetrate the blood-brain barrier. So blood-brain barrier issue, I don't think this can be translatable. So now the question is, we asked that what is EGS2 PRC complex is controlling that, giving this plasticity and therapeutic resistance. And we did a simple experiment, knocking down EGS2 and hit them with temozolomide. Asked the question is, what gene is regulated? And one of the top gene came out is this ARL13B, which is a ciliary protein that regulated during TMZ therapy. So ciliary protein is a sensory organelle that expressed by many cells, and it is responsible for very key signaling pathway like sonic hedgehog. And ARL13B is stabilized these structures and also responsible for cargo movement. It never been investigated in GBM, and what you can see is that during TMZ therapy, the ciliary structure is elongated. Not only that, if you knock down ARL13B, you can significantly prolong the survival of animal and block the glioma growth. This is very interesting because at least in vitro, these cells grow absolutely fine, no problem at all. but in vivo, they cannot engraft anymore. So now we have shown that the EGH2 epigenetically binding this and regulating this ARL13B protein. I'm not going to go into detail, but the question is, what is ciliary protein doing it? So we did a mass spec analysis, pulled down ARL13B and did the mass spec during TMZ therapy. And what we're able to show that it interacting with a very important protein called IMPDH2, which is a de novo purine biosynthesis rate limiting enzyme. IMPDH control this de novo purine biosynthesis, and purine is very important for metabolism of DNA and RNA. And to follow my talk is a very complex process. You have to know two things. Purine can be synthesized de novo from the scratch ribose 5-phosphate, or purine can be recycled from the high energy molecule like RNA, DNA, ATP. In brain, and the purine salvage is predominant because it requires less energy. So they recycle purine. Okay, so now the question is why these cells try to regulate this purine during TMZ therapy. And we showed that ARL13B is interacting with IMPDH activity, IMPDH2. Now question is why ARL13B interacting and what it is doing it, okay? So what we believe is that the purine recycling and purine salvaging is very important because as we know, the TMZ alkylate this purine. So now we could ask the question, what is ARL13B doing? So what we did is like we knocked down ARL13B and do the radio level tracing. And what we're able to show here is that, and then we track this radio level in the DNA and RNA. And what we're able to show that, and we all did all this thing, but quickly what we're able to show that, that if you knock down ARL13B, what happened is that this purine salvage is unleashed. So both RNA and DNA, and what we believe that what's happening is that ARL13B interact with IMPDH2, block this purine recycling, and force the cell to switch to de novo cycling during TMZ therapy. De novo, remember, they're making the fresh purine, okay? So why cell wants to do that? What we believe that during TMZ therapy, because this is purine is alkylated, if you recycle this purine, cell get damaged, okay? The DNA will get damaged. So cells somehow sense that. That's why they switch to purine de novo synthesis where they make the fresh purine. And we have shown that in the experimentally, that this is exactly what's happening, is that this O6MZ get incorporated in the DNA if you remove ARL13B, and it's shown by mass spec. So the summary is this, is that take-home message is that ARL13B is regulated by epigenetic modifications, and that leads to blocking of the salvage pathway, and that's unleashed this de novo pathway, thus protecting the cells from this using the damaged purine and DNA damage. And now question is, how can you translate this in animal, in patients? So actually purine can be, this pathway can be targeted by a immunosuppressive drug called mycophenolate mofetil that has been in the clinic already for transplant setting, okay, for kidney transplant. So now when you combine this with temozolomide, what we find is that these become more sensitive to TMZ. Okay, and we can completely protect this animal for developing tumor when you combine this MMF, mycophenolate mofetil, with TMZ. Based on this, we launched a clinical trial. Now we recruited almost all the patients. Now we're analyzing data. We're very excited about this. And then we'll publish the data pretty soon, but their data looks super promising, no side effect. Patient tolerate very well, and we are also receiving some nice signal that might justify you combining this MMF with TMZ for glioma therapy. So I think I'm gonna stop here. So summarize my talk is that I'm sorry I went a few minutes over, but interconversion between differentiated glioma cell to dedifferentiated cells as a mechanism of resistance. And we show this mechanism of resistance is driven by epigenetic plasticity. And that leads to this blocking of the purine salvaging and unleashing the de novo purine salvaging. And you can now block this, this de novo pathway by MMF, which leads to sensitivity to TMZ. So thank you for your attention. I'm happy to ask you, give you an answer any questions you have. And I appreciate this opportunity for presenting our talk. Thank you.
Thank you very much, Atik. Very interesting speech. Maybe I start so the people, if they have a question, they can ask later. So you show us that the way of escaping of the glioblastoma cells is like the differentiation to the stem cells, that the mechanism behind this differentiation is like the changing in the metabolism of purine. And you show us also that you perform some trials with the drugs that blocks the de novo synthesis of purine, and together with telozolomide. So my question is, in the patient, you didn't have some effects like immunodepression, because all those drugs that are used for in the block of purine normally are used to downregulate the activity of the immune system. So you haven't noticed that in the patients?
So that's a very, so that's a very important, good question, because the question you're asking is that, okay, MMF is an immunosuppressive drug, and TMZ also we know is an immunosuppressive, yes, myelosuppressive. So when you combine them, you should have more severe immunosuppression and patient gets sick. Very interestingly, what we see is that with respect to immunosuppressions, that dominant factor is TMZ. So if you add top of MMF, they don't, the patient don't suffer more immunosuppression, which is supposed to, but actually the immunosuppression patient in this case is driven by TMZ. So adding MMF, we are now, I think the maximum tolerated dose, which is 2000 milligram per BID. I think this is the last cohort of patients. So far, no toxicity. Patients are doing great.
Yeah, sounds really good, really promising.
Yes. So it is about we are at 25 patients. None of them show any dose limiting toxicity.
Okay, thank you very much.
So regarding that, one argument that people make, especially at Duke, is that immune suppression, that myelosuppression can actually be utilized in that when you've got that gap, when the big drop in leukocytes, there's a rebound effect. It's called homeostatic proliferation, whatever the hell that means. But the idea would be that you could then sort of vaccinate into that hole, and the cells that come out of it are actually more, they're less immune suppressed and they're more easily stimulated. So I'm wondering, have you tested any of this in an immune competent model to see if there was a way to sort of combine an immunotherapy with it? Because it seems like immune suppression may be exactly what you're wanting to do, is suppressing the appropriate cells and allowing the cells you want to respond to come out of it.
Yeah, exactly. So yes. So you're talking about the Duke idea of that. So what they do is like create space in the immune compartment, but depleting the cells, pre-existing cells, and then expanding the re-educated immune cells in the compartment. No, we did not try, Michael, because that get really difficult to get approved by the FDA, because this is a first in patients trying, so we're not thinking that way yet. we want to see this, but that's a good idea. Yes, I think that's a logical step of combining this with the PD-L1 and see whether you can re-educate the immune system, because you're going to have a more compartment space in the patients.
Well, and if I can just dominate the conversation here and throw more questions out. So years ago, Heidi Phillips showed that when everybody was all about taking the stem cells and showing that the stem cells, when you put them intracranially into nude mice, it took 100 cells to turn into a tumor, and the non-stem cell, whatever they chose to call those wouldn't grow. But she actually showed that they would grow. It would take months for them to come out. And so I was kind of wondering about that dedifferentiation phenomenon that might actually, this seems to just sort of affirm that there is this kind of dedifferentiation that can go on. But regarding the fact that the going through the de novo purine pathway versus the salvage pathway, I think one thing we need to bear in mind is that due to lots of weird things that go on with the purinergic receptors on tumor cells, there's a lot of ATP in the extracellular environment. So tumor cells have almost a glut of ATP to absorb, and they'll eat this like candy, and it doesn't matter where it comes from. So I think that the de novo pathway is not a problem for the tumor to carry that out.
Exactly. So you're absolutely right that they have a lot of extra the metabolite in the microenvironment to feed them. Yes, and you show that they switch to lipid metabolism. Yes, even to produce the energy. And we find the same things when cell de-differentiate. Yes, into cancer stem-like cells, they also switch to lipid metabolism, and they prefer lipid metabolism over any other metabolism. So we still don't know why it is happening. But going back to your ATP, is that one of the things that we're struggling is that, this salvage pathway and this damaged nucleotide, how the cell sensing this? Yes, this purine is now alkylated during temozolomide. That's where they're switching from salvage to de novo. That's our hypothesis. But one thing we're struggling to understand, the sensor for this alkylated purine, that how the cell, it's kind of boggled my mind that cell can sense this simple change of this one alkylated purine and then switch suddenly to de novo pathway. So I don't know. I don't have the answer for you. Yes, there are lots of ATP there, but how the cell sensing that, I don't know. I don't have the answer.
That's an excellent point. And I'll stop after I say congratulations for getting this moved into the clinic. That's just awesome. Yes,
it is. As a basic scientist, it's probably most gratifying thing for me, seeing, because it took us almost 6 years. But it's in the clinic and it's almost done. So we are very excited about it, because at the end of the day, we get caught up with this idea of publication, grant, and this and that, and forget of the real purpose that we hear is about for our patient. Yes. And as a basic, sometimes we kind of get distracted about this from all this noise.
Well said.
Yes, I think one more question.
Thank you, Ahmed, for your nice talk. Regarding your last proposition, how the cells sense and how they make decisions. From bioinformatic point of view, I think the cell communicate. They are always in the communication. So probably some network analysis and how they communicate by the groups, like at the single cell level, and how they communicate and what ligands and receptors are affected will help you to answer this question. But
no,
of
you're
course,
absolutely,
you need to have.
Yeah, you're absolutely right. I think now we have the tools to study the single cell analysis. I think more than single cell analysis, I'm very excited about this spatial transcriptomics. Yes, even though we are not getting the single cell resolution, but I think we should get it, and we are getting some exciting data to understand how the cells are communicating with this single cell spatial transcriptomic analysis. And we are getting some idea, but we're still not there. But you're absolutely right that how the cell communicate, they live as a community, and we still don't know their language. Unless we know their language and how they're communicating, I think it will be very difficult to treat this disease. And we are getting there, but I think you're right, absolutely.
Exactly, you are right about the spatial transcriptomics too, because this will give you the idea of the position of cells in space, where they are located precisely. And this is an advantage to do spatial transcriptomics and then to look at the communication between those cells and group of cells.
Yes, I agree with you 100%.
Thank you very much.
Thank you.
Are there any other questions from the public? Okay, if not, we pass to our last speaker, Iris Chiara Sararoglio. Iris, if you can put your presentation.
Iris, we cannot hear you. Probably you are muted.
Okay, we always do not hear you.
Now
Iris,
you can hear me?
yes, now yes.
Yes, okay.
Iris,
Perfect.
can you put full screen?
Yeah, of course.
Okay, perfect.
Okay.
Yes.
I'm sorry because I hear my voice, so I try to modify the audio. Just a second. Why? Okay. I don't know.
Try to test something.
Okay, it doesn't matter. I see my voice. I hear my voice. Doesn't matter.
Sometimes,
we hear you twice.
Sometimes if your phone is close to your computer, that might echo it. So can you, is your phone is close to your computer?
So, okay.
Se hai il telefono vicino forse ti rimbalza.
Sorry. No. Okay. Here you can hear.
No.
Why?
I think also if you might be close to each other, you might have the echo as well, if you are in the same room or something.
I don't know why. We try here. You can hear me?
Yes.
Yes, better.
Okay. Okay, it's better. Perfect. Also for me. Perfect. Okay. So thank you for this invitation. And today I will talk about how glioblastoma can modulate the blood-brain barrier permeability. So as you talked before, glioblastoma multiforme is one of the most aggressive and lethal tumor of the central nervous system. It is an incidence of 3 new cases for 100,000 people per year, with a median age between 60 and 70 years old. The treatment provides surgical resection, follow radiotherapy and chemotherapy based on temozolomide. It is the only drug that can cross the blood-brain barrier. And this is used as a first line treatment of glioblastoma, but the prognosis is very poor, as the other speaker said before, due to the tumor heterogeneity, DNA damage repair mechanism, and the presence of cancer stem cells that have intrinsic characteristic of chemoresistance, and also high expression of ABC transporters that exclude drugs from tumor cells. And these cells cause local relapses because they escape from surgical resection and spread in the surrounded area of central nervous system. Another obstacle to the success of chemotherapy is the blood-brain barrier, the strict endothelial cell by adherent junction and by junction, and the presence of ABC transporter limits the drug delivery. So in this, it's known that blood-brain barrier in presence of glioblastoma is disrupted. And this is disrupted in a different way. So is completely disrupted in presence of the tumor bulk, but partially or not disrupted in presence of infiltrating glioblastoma cells and cancer stem cells. Inflammation is related to the cancer, and the glioblastoma microenvironment is rich of cytokines that can induce tumor growth or tumor suppression. And during the inflammation, obviously, blood-brain barrier cells are exposed to pro-inflammatory cytokines that can induce a changing of permeability, BB permeability. So in this context, our work is to understand how glioblastoma cells can modulate the BBB integrity, and in particular, if the grade of differentiation of the stemness of glioblastoma can influence the BB integrity and drug permeability. To this reason, we develop in vitro model of glioblastoma and blood-brain barrier in order to understand, to answer this question. So we used the human brain microvascular endothelial cell line, D3, that retains in vitro the feature of a competent BBB, and cells derived from primary surgical specimen of glioblastoma patients. So from this specimen, we isolated the cancer stem cell, called here neurospheres, and the differentiated cells called adherent cells. And as you can see, the neurospheres show characteristic of cancer stem cells with self-renewal, clonogenicity, and vivo tumorogenicity. The differentiated cells, no. So we set up a co-culture model in which we seeded in the upper chamber BB cells, and the lower chamber, the glioblastoma cells, either neurospheres and either adherent cells. And the second co-culture model, in which we seed the BBB cells and only the medium obtained from glioblastoma cells. And the first result that I show you is about the permeability, the blood-brain barrier permeability to doxorubicin, that is a substrate of PGP. So an index used as an index of PGP activity, mitoxantron used as an index of BCRP activity, and the trans-7 Fitch used as an index of tight junction integrity. And in our model, we can see that the glioblastoma cells induce a more permeability to doxorubicin, mitoxantron, and trans-7 Fitch on BB cells. In particular, the differentiated cells induce more permeability to these substrates. The interesting thing is that the only medium conditionate, so obtained from glioblastoma cells, produce the same effect, and in particular, the conditioned medium obtained from adherent culture induce more permeabilization. After this result, we investigate the ABC transporter and tight junction expression on blood-brain barrier cells. And as you can see, the adherent culture, the adherent cells induced so a lower expression of PGP, BCRP, occludin ZO1 in BB cells. Also the neurospheres in a lower way. And this modulation was present at a preterine level and also at the transcriptional level. Also, the conditioned medium induced the same effect. So a reduction of ABC transporter and tight junction expression. Okay, so since medium produced the same effect of cells, we think that soluble factor released from glioblastoma cells can modulate the BB permeability. So we set up a cytokine array on conditioned medium, and we saw that IL-6, IL-8, MSCP1, and TNF alpha had different expression between the two media. So we confirmed this data by ELISA assay, okay, in a quantitative way, and only IL-6 show a a difference between these two media. In particular, there was a higher expression of IL-6 in conditioned medium obtained from differentiated cells. And so to confirm, to demonstrate that the IL-6 could be modulate the blood-brain barrier, we set up a complementary experiment in which in the first we seeded the blood-brain barrier cells, and the conditioned medium obtained from neurospheres that is poor of IL-6 added with IL-6. And in the complementary experiment, we seeded the BB cells in presence of adherent medium that is rich of IL-6, in this case is depleted of IL-6. And as you can see, the neurospheres medium plus IL-6 show the same permeability, increasing permeability of the differentiated medium, whereas the differentiated medium depleted of IL-6 show the same permeability of the neurospheres medium for all substrate, doxorubicin, methoxylandron, and destran. As a proof of concept, we set up this co-culture. So we seeded the BBB cells with a fresh medium, added IL-6. And as you can see, IL-6 induces a more permeabilization of a blood-brain barrier. So at this point, we think IL-6 could be a good candidate that modulates BB permeability. And now we are investigating the mechanism by which IL-6 can act on BBB cells. And we know that one of the downstream signaling of IL-6 is that JAK STAT3. And that the JAK and also STAT3 can modulate ZO1, Claudin-5, and Claudin-3. In our preliminary data, we can see that the differentiated medium induces an upregulation of the JAK-STAT pathway, and the differentiated medium depleted of IL-6 induces a downregulation of this pathway. Also, the differentiated medium change the level of occludin expression, the tight junction expression, so the one, Claudin-5 and Claudin-3. Obviously, this data has to be confirmed with a complementary experiment using neurospheres conditioned medium, in this case, enriched by IL-6. On the other hand, STAT3 can modulate the ABC transporter expression like PGP and BCRP by the arylic hydrocarbon receptor. And so our idea could be, well, our hypothesis is that IL-6 can modulate these ABC transporters by this transcriptional factor. So in conclusion, in this study, I show you that the grade of differentiation of glioblastoma cells influence the permeability of BBB, and in particular, the differentiated cells that induce a lower competence of BBB, so a more permeability. Glioblastoma cells decrease the tight junction and the ABC transporter in blood-brain barrier, maybe inducing the release of a soluble factor. And among these soluble factors, IL-6, mainly produced by differentiated cells, is a good candidate that can modulate BB permeability. And modulating IL-6 level or IL-6 signaling BBB cells can be a new tool to improve the delivery of chemotherapeutic drugs in the glioblastoma tumor, and maybe eradicate glioblastoma cancer stem cells that are not removed by surgery. I would thank my lab group, in particular my boss, Regante, Johanna Kopeczka, our collaborator and grant that made possible this work, and all of you for your attention. Thank you.
Thank you very much, Iris. Are there any questions? Maybe the topic is a little bit different than the other ones because we speak more about the blood-brain barrier than about the tumor, but this is very important because also, as Atik tell, it's very difficult to find the drugs that can pass the barrier to improve the results of the treatment.
Yeah, I have a question for you, Iris. So my question is more kind of philosophical than. So why you think that tumor cell wants to create a permeable BBB? I mean, isn't it go against tumor survival, or you think that pro survival? Because I'm just, I mean, you think about a lot about this more than me, but I'm just trying to understand, if a tumor is there, and if the drug's coming in through blood-brain barrier, and the blood-brain barrier preventing the drug to comes in, why you think that tumor wants to make that blood-brain barrier permeable? Do you have any idea on that?
Okay, a good question, thank you. I don't know. We have this evidence, and in the literature there is evidence that the blood-brain barrier is disrupted in presence of the tumor. But obviously, when the situation is very compromised. So now we are trying to understand why and in which way this happened. I don't know is a pro-survival or not for the tumor, but is an evidence. And so we are studying why success.
Now,
yeah.
Go ahead, please.
No, and we found that IL-6 could open this induce more permeability, but obviously for the clinic we have to put in the right. We have to set and put in the right condition, because obviously if we have to administrate IL-6 in systemic way, I don't know, I think could be dangerous now. So maybe it could be possible to administrate and in a formulation and tissue and targeting the tissue. So
So now, did you test that in vivo? What happened? So you're showing that IL-6 is produced and they are permeable, but maybe there's one experiment that will be very fascinating, is that if you knock down IL-6 in tumor and then implant that tumor cells into the brain of the animal and see how the blood-brain barrier is affected in that, because what you're showing in vitro may be completely different in vivo. Yes, so you need to because the microenvironment it's completely different. Yes, so I think it would be nice to show that.
Course.
And
yes,
then,
of course.
And then one thing I want to show you that I don't know, we published one paper a few years back that under temozolomide, these cells, GBM cell express IL-8, tremendous amount of IL-8. Okay, and IL-8 and IL-6 can induce transdifferentiation, not dedifferentiation. Transdifferentiations of glioma cells to endothelial-like cells. Okay, these glioma cells become endothelial-like cells. So I wonder there is a connection there. I don't know. I'm just.
Okay. Good.
Something to think of.
Good evidence. Yeah, of course. I think it's important to set up in vivo experiment, to see exactly what happens, because vitro and in vivo models are give different results. No.
Yeah.
Well, I'd make an argument that the tumor, first of all, might not really care that much about the blood-brain barrier. It develops its own intrinsic mechanisms of defense. And this may just be a consequence of having an inflamed microenvironment. So the neuroinflammation or the CNS inflammation that occurs, driven not just by the tumor, but also by the tumor microenvironment cells, who will also contribute to this. I think the IL-6 knockout would be really curious to see, is that complemented, or is it sort of compensated by the endothelial cells, by astrocytes, by other cells in the microenvironment? I think that's an actually fascinating idea. Obviously, people are targeting IL-6 for a number of reasons in a variety of diseases, including like craniopharyngioma. And I always worry about that because it's such a pleiotropic molecule that are you going to make things better or worse by dealing with that? But and again, living in the exosome-centric universe that I live in, I might argue that opening of the blood-brain barrier would allow extracellular vesicles to be released more easily. They cross the blood-brain barrier just fine, both directions. But from say an immune suppressive mechanism, if these were able to get out into the peripheral circulation and encounter T cells before they ever got to the tumor, they could actually sort of disarm or disable those T cells. And that's just my hand waving view on that. But this is a truly fascinating study. And I think there's an awful lot to go on here and amazing directions to take it.
Okay.
Are there any other questions for Iris?
Iris, you are a postdoc or graduate student?
Postdoc.
Postdoc. Congratulations on your great work.
Thank you so much.
So I think we can tell that we finish with our presentation. I don't know, Lori, to you, the meeting.
Okay, thank you so much, everyone. The last one, I will introduce our journal. Wait a minute, I will share my screen. Can you see my screen?
Yes, we can see it.
Oh, okay, now I will introduce cancer drug resistance. Our editor-in-chief is Godfrey Peters, and this is our editorial team. Susan Song mainly take charge of the journal management. And Laurie Kang, I take charge of the special issue construction and the webinar interview. Blanda Zhang is responsible for manuscript processing, and Bella Zhao is responsible for conference cooperation and promotion. So if you have any questions, please contact any of them. They will solve your problems. And our journal is an international peer-reviewed open access online journal, and the journal publishes research articles, reviews, case reports, commentary, and letters on pharmacology aspect of drug resistance and its reversal, including drug design, drug delivery, drug distribution, and ancillary drug resistance. And our journal launched March 2018, and up to now we have 318 publications. And we also cooperate with many international partners. You can see here. Regarding the indexing, we have indexed by many database, which is in the right blank. Particularly this year in August, CDR is indexed by EICA, and next year we will get the first impact factor. Also, to CDR get this achievement without the editorial board members. So now I will introduce our editorial board distribution. There are 67 editorial board members from 17 countries, and we can see mainly from United States, Germany, Italy, the Netherlands, and things like that. And most of the expert they're each indexed from 40 to 60. And the junior editorial board distribution is at the same. So with the editorial board members' help, they control the high quality of our articles. So we can say the high set articles published from 2020 to 2022. With the help of the editorial board members, we can say most of the articles from CDR get most citations. In order to get a higher achievement, we do a lot of change this year. We can see from the homepage. From the homepage, if there have some big events, and we will update in the center of the homepage. And at the bottom of the homepage, there are some data, and also this data is updated in time. Now we can see the articles. Mainly, it's easier for us to download the PDF version. Particularly our journal is encourage the expert to provide the geographical abstract, which is used to attract many readers. And from this year, September, we start to hold online webinars to enhance interaction. And up to now, we have 3 completed webinars and 2 ongoing webinars. And you also can check the ongoing webinars on our homepage. And also from July to September, we conducted 6 expert interviews. We will continue to conduct interviews with experts to cooperation between expert and editorial team. So if you have the new articles, or if you have some advanced research outcome, welcome to we will interview. We will take the official interview with you, and in order to promote your new research. In order to get more promotion, we have a YouTube account, LinkedIn, and Twitter account. So if you have this account, please follow us. And for today's presentation, we will post your presentation on YouTube. So if you have some unpublished data or privacy data, please tell me, and I will not publish your presentation online. Okay. Lastly, the 2020 Best Paper Awards has already been set and will announce next year on January. We will launch the 2021 and 2022 Best Paper Awards successively. So if you have suitable papers, welcome to submit to our journal, and we will get the first impact factor next year. So this is all the introduction, and thank you for all your participation and great support. Thank you.
Thank you.
So do you have any questions? Okay, so now I will put your presentation online. Okay.
Laurie, if you can, the presentation of Iris contain the data that we are still finishing the last experiment, so they are not published yet. So I would prefer that they are not available online, because
okay.
That's okay. Thank you.
Okay, got it. So I will not publish your presentation. So Michael and Antique, can I put your presentation online?
Yeah, you can post mine. Those are all published data. So
okay, thank you. So how about Michael?
If anybody wants to steal my data, they're stupid. So sure, go ahead and put it out there.
Thank you so much. If you don't have any questions, so this is our webinar. Yes, thank you for your great support.
Thank you so much.
Thank you.
Thank you.
Bye.
Bye.
Thank you.
Bye.
Bye.
Madonna, questa roba io no.